Hello guys, this is Zoologist AK and I'm here to talk about gene regulation in case of prokaryotes. And in this video, I will talk about Operon, that is arabinose operon, that will activated only when arabinose as an substrate present in the culture medium or the environment in which bacteria, that is E. coli, is growing. And this Operon or arabinose operon exhibit much more complex pattern of regulation. That is, in this operon, the regulatory molecule exhibit both positive and negative regulatory effect on the transcription of different structural gene in case of Operon. But this Operon is not completely understood yet, but whatever discovered about it, we are going to talk about that in this video. And this diagram represent you the structure of this Operon. Here you can see these are the structural gene that code for different enzymes, that are D encode for L-ribulose 5-phosphate 4 epimerase, and A encode for L-arabinose isomerase, and B encode for L-ribulokinase. And these enzymes catalyze the catabolism of arabinose into D-xylulose 5-phosphate. Here you can see in this diagram the mechanism of action of these three different enzymes that are secreted by three different structural genes of Operon. Arabinose is converted into L-ribulose by L-arabinose isomerase, and L-ribulose is converted into L-ribulose 5-phosphate by L-ribulokinase, and this L-ribulo 5-phosphate is converted into D-xylulose 5-phosphate by L-ribulose 5-phosphate 4 epimerase. So this is the mechanism of action of these three different enzymes. And coming back to the structure of Operon, here we can see this C. It is the regulatory gene. It acts as a repressor and inducer both. In the absence of cyclic AMP and in the absence of arabinose, that is the substrate for E. coli, then this C encode in protein C protein that act as a repressor molecule. But in the presence of cyclic AMP and in the presence of arabinose, that is the substrate molecule, it act as a inducer molecule. And this C has its own promoter, that is PC, that regulate the expression of this C gene. And here you can see this BAD, it act as a promoter gene for these three structural genes that are present in Operon. And this I here is represent here the inducer gene, and this cap is our cyclic AMP and and our cap binding site, that is our catabolic activator protein. And this O1 is represent here the operator site at which RNA polymerase will bind and will activate the expression of these three structural genes. And at this operator 2, that is represented by O2, this is the binding site for the protein that is secreted by C. So the protein that is secreted by C will bind to this O2. So this inducer gene, that is I, it consists three binding site, one for C and another one for RNA polymerase that will do transcription of different structure genes, and cap binding site that will bind in a complex form with cyclic AMP. This cap has two binding site, one for DNA binding site, and another one is cyclic AMP receptor protein binding site. So it bind to the cap binding site of this ERA gene only in a complex form in which it is bind with cyclic AMP molecule. And this ERA-C act as a regulatory molecule when, and in case of regulatory molecule, it exhibit both type of mechanism, positive and negative. In positive mechanism, it will activate the transcription when arabinose and cyclic AMP will present in the substrate medium, and this arabinase will bind to this C repressor protein, and repressor protein that is present in dimer form, and will result in conformational change of this C repressor protein, and due to which this C will unable to bind to O2, that is operator for this C protein molecules, and the cyclic AMP will bind to the cap and result in promotion of transcription of different structure genes by binding this RNA polymerase to the O1, that is O1 operator 1, where RNA polymerase is going to bind. Here two RNA polymerase binding site is represented. One is promoter BAD and another one is O1, that is operator 1, and here is the promoter for different structural gene B, A and D in case of Operon. This diagram represents the Operon in repressed state, and this repressed state occur when there is the absence of arabinose in the as a substrate molecule in the culture media, and there will be absence of cyclic AMP. Here in this picture you can see that C as a dimer bind to both sides O2, that is the operator and binding site for this C protein molecule, and another one is I, that is I gene that encode for inducer molecule. So this C bind in a dimer form to both O2, that is operator 2, and I, that is inducer. And as we know, inducer had three binding site for polymerase, cap and this C. So it will block the transcription of different structure gene, because the dimer binding of this C molecule to O2 and I result in formation of a DNA loop, as you can see in this diagram. Due to the formation of this loop, RNA polymerase cannot bind to the promoter BAD. This promoter is for gene B, A and D, because this structural gene BAD express cooperatively in a single mRNA polypeptide chain. So here this loop formation inhibit the transcription of different structural gene in case of Operon, and it is caused by C when there is the absence of arabinose and cyclic AMP in the culture media. And this picture represent you that Operon in induced form, means it will induce the transcription and then translation of these three structural gene into their enzyme molecules. Here in when Operon is in induced state, the arabinose C protein and cyclic AMP and cap complex both bind to their I site. As you can see here, this is cap and cyclic AMP, and here is C, which is bind with arabinose at their binding site, that is present in I gene that will encode for inducer, and due to which this loop formation does not occur, and this RNA polymerase able to bind to the promoter BAD, that is the promoter for gene B, A and D, and result in the transcription of all these three gene, and then translation of these three gene result in the formation of different proteins in a single polypeptide chain. So this was all about era Operon. If you like my video, then please subscribe to my channel and press the bell icon that is shown below, and please do like, share and comment. If you have any query, then please write it into comment in the description box given below. Thank you for watching.